Good morning. Welcome to New York. My name is Ed. I'll be one of your hosts this week. And we hope that you are going to have a wonderful week learning about tomography, as well as learning a little bit more about what we do here in New York. A few housekeeping things. We like to thank our sponsors, Sub Angstrom and Thermo Fisher. Without their support, we wouldn't be able to have this short course. And we thank you also for coming here. Something that we'll mention a little bit later is where are we? We're in New York. We're at the New York Structural Biology Center, and we do more than just EM. We focus on all three modalities of structural biology, including NMR, X-ray, and EM. And a little bit later during the lunch break, if you want a tour of the facility, I'll also give you a tour. Something that we have beyond high-end instrumentation is a lot of people. So throughout the week, the main reason why you might notice we have as many instructors, TAs, and helpers as students is that we realize 1 week will not make you an expert in electron tomography. It's mainly to give you small vignettes to really create that network and give you the ability to learn more. And something that I really want to highlight is that there are a lot of people behind the scenes and throughout the week that you'll be interacting with. And the best thing to do is not just sit through a 30-minute lecture, but actually talk to the instructors, talk to the TAs, and really create that network to learn more. And that's really the purpose of bringing you here on site. And something that is even more amazing is usually all your fellow classmates become a cohort and colleagues and another resource. So you might be going back to your home institution. You may or may not have a cryo-EM expert, a structural biology expert, let alone a tomography expert. But hopefully, as all of you go through that journey, you can help each other. And they'll also be here as a resource. As I mentioned, we have a lot of instrumentation, and during the tour, we can see a little bit more of that. Overall, NYSPC has 9 Krios instruments, 4 Glacios instruments. We also have a whole host of sample prep equipment, and some of them you'll be able to see demos of throughout the year. I want to point out that how we started was really through the NIH Common Fund. The NIH Common Fund created national centers like ours, and what we were trying to do is not only lower the barriers of access to cryo-EM, but enable that everyone can access cryo-EM. And part of that is that we have a network of EM centers. So yes, we're here in New York, but there's a network of 7 service centers and curriculum development centers. So not only do you go to certain places to make use of instrumentation, but there are a lot of learning materials online. So I would like to point out that the 3 cryo-EM centers proper are in New York, Portland, and Stanford, but we also have a cryo-ET network focused on the hub based in Madison, and we have 3 other satellite centers in Boulder, Stanford, and as well as in New York. We will be using quite a bit of our online materials. Something to note that by being here, we're going to be recording, we're going to be taking a lot of photos, and hopefully if all of the lecturers do post their slides, they'll be on the website as soon as possible. To help with communication, I'll be using the Slack channel. I know many of you are on there. How many people have not been able to access Slack? Okay, good. So we'll have as many updates as possible. All the photos that I say we'll be taking will go in there, and hopefully that will also be a resource. Our NYSPC NCCAT workshop workspace is a pro Slack workspace. So even after the course, you can still be there and you can still ask questions from not only our staff, but the instructors. So what we're going to be doing today, how the short course is arranged is that the morning we'll primarily have lectures up here, and then in the afternoon we'll have practicals. In particular, today, I don't want to cut your lunch too short, but around 12:30, if you're interested for a tour, I'll be leading a tour and we'll do a walkabout NYSPC. You can see some of the instrumentation. So again, welcome to NYSPC. I want to try to keep this short because I'm not really the main star. I'm just the host. The instructors and I guess you as the students are really the focus of this week. So hopefully we'll get to know a lot more about each other. And again, welcome to New York. Okay, so Wei, do you want to help set up? You can kick me out. So our first speaker will introduce tomography, and our first speaker is Wei Dai coming from Rutgers. What is your favorite piece of IoT instrumentation? Good. Okay.
The artist.
The artist, we have one. Unfortunately, we just signed off last week, so you'll be able to see a little bit more on a walkabout. And I see Jeff clapping her hands because we have a huge thermal presence. Do you know how to use the art?
I could point it towards you.
Yes. Okay.
What do you like this?
So I just need to share my
You
screen.
just have to share it. You can continue.
So everything recorded.
Everything should be recorded, right, Adam? Okay, and if you need a laser pointer, it's right here. Thank you.
Okay, so I'm really happy to be here from New Jersey Rutgers University across the rivers. I'm really happy to be here to give the opening talk on introduction to ground electron tomography. So actually, there are more people than I expected from the list Ed sent to us. So let me just get a really quick survey. So let's say there's 3 levels of tomography. You never use it, never heard of it, and then basically level 0, no knowledge, no practical experience. Level 1, you kind of heard of it, or maybe you see other people doing tomography, or at least read a couple papers on tomography. Level 3, you somehow get your hands on tomography applications. So who is level 0? Basically, totally beginner. Level 1, okay, level 2, and there are people never, okay. So basically, you know level 0, they're half level 1, half level 2. So what I'm gonna do, this is the learning goal for today. So it's called Introduction to Electron Tomography. So what I'm gonna do is I'm gonna share examples to give you an overview of the history, the challenges, the application of ET in structural biology. So this is pure discussion. I'm gonna lead the discussion, and our focus will be how advances in instrumentation, in computational structural prediction, in sample preparation that expand ET application in cell biology. And then by end of my talk, I'm gonna share my research as one example to show you the workflow of a ET project, and then how we combine complementary approaches, including biochemistry, biophysics, proteomics, with ET to understand molecular landscape of fungal plasma membrane proteins. So biology involves structure of different sizes, and there are lots of imaging modality for us to able to understand their structures. So TEM-based tools can be separated up into 3 classes. So single particle analysis, micro ET, and crowd ET. And then many of you, probably more of you, will have experience with single particle analysis or micro ET. Basically, those are the 2 tools to give you atomic resolution structure of your favorite protein complexes. Crowd ET really stands out as a powerful tool for you to be able to understand structure of your protein complexes in their native states. Okay, so what is crowd ET? And then this is a video I got from YouTube. Your favorite sample here is basically a part of a cell applied to EM grid. Just like doing CT, you take images from different direction and then computationally back project them to get the 3D map. And you can annotate them, basically see all your favorite protein complexes in the cell. So I always show this graph to show application of crowd ET in cell biology. So I think if you look at maps currently deposited in EMDB, so tomography application is still catching up. So the maps currently in EMDB is far less compared to maps deposited from single particle analysis. And then I'm trying to go to EMDB to trace back when is the beginning of crowd ET application in biology. So it's actually pretty challenging. The very first map deposited by tomography application that I was able to find from EMDB is actually the tomograph of thin section plastic embedded insect-like muscle cell from Kim Taylor's lab. So they also performed subtomograph averaging, cutting out small pieces, and they were able to fit atomic models of the Rutgers cross bridges into the subtomograph average, and also apply the real space refinement to polish, to refine the fitting. So I understand this is a tomograph of chemically processed sample, but still think about the concept. You combine high resolution structure of atomic models with your lower resolution tomographs to get a holistic understanding of the domain organization and subunit interaction. This concept is still very applicable to many projects, even people are doing at this time. So, actually the tomography application in cell biology entered the cryo stage or the ice age a lot earlier than I expected. So this is a tomogram, cryo-electron tomogram of the periphery region of the dictyl stadium cells. So they basically, I think for this project, they were really smart picking up this particular sample because we know that this cell really mobile. They can really spread out, forming a very thin layer on EM grid to allow clump freezing and then tomography. But even that, the tomography study is still limited to the peripheral region of the cell just because limitation by the cell thickness. And then by doing this, they were able to resolve a very nice network of acting filaments. So I feel this is really a milestone in cryo-ET application because it's one of the early efforts in tomography in cryo conditions. And also this is the study of the acting network in intact cells in absolutely native state. Okay, so next one. So we can kind of say, cryo-EM single particle analysis started from phages and various particles. That's because the axial heterosymmetry reduced the number of particles required for, reconstructing high resolution maps. So in the early stage, early 2000, so cryo-EM single particle analysis can already push resolution to almost atomic resolutions. But this particular study, they applied cryo-electron tomography to the whole HSV variant. So basically, we know quite a lot about packaging of the capsid because development in single particle analysis. But if we look at the entire particle, including the envelope and also the tegument protein, we know very little about the tegument layer or the envelope because they're not symmetric. They don't have symmetry. So during the application of symmetry, you lost those components. So in this particular study, they were able to resolve the tegument layer in intact HSV virions. So although the tegument layer is somehow polymorphic, they still resolve it as a cap shape density, usually distributed to one end of the envelope. And then they also perform the subtomograph averaging and then focus on the analysis of the pentamers. And then they were able to show actually I can't see my screen over there. The pentamer at the distal end of the tegument versus the proximal end of the pentamer, they actually have different density associated with it. So this example I picked up basically to show tomography, although the resolution was a little bit limited at that time, it can complement single particle analysis to give you information of the elements that is polymorphic in your biological samples. So in my postdoc study, I was assigned a project to study phage-host interaction to understand the phage assembly process. So if you're working on a microbiology or virology project, when you purify phages, so usually you enrich the final product. It's very hard to capture the intermediate. Even you always doubt, maybe that's just deformed the particle during my purification process. So to approach this project, I decided to use tomography, basically directly image the phage infected cells instead of purifying particles out of the host. By doing that, I was able to resolve structures of the intermediate, the baby phages rebuilding. So even the phage, the host, actually cyanobacteria I pick out, is a relatively small one, in the range of maybe 1 to 2 microns. But the thickness is still a problem to get better view of the baby phages. So this project was possible because of 2 factors. One is the infection process actually generated the leaking cells. So cytoplasm will be leaking out during the infection process, especially during the late phase. And secondly is the application of the phase contrast optics, which dramatically improve the resolution through the contrast of images of the tomographs. We were able to clearly identify those baby phages in the very crowded cellular environment. So next one, starting from early 2000 until maybe around 10 years ago, 2015, we have a lot of people working on crowd-ET to study organization of protein complexes from cellular fractions or kind of enriched or purified organelles. This is one example by crowd-ET tomography of mitochondria fragments. You will be able to resolve the very unique packing of the cristae membrane vesicle and also the 2 rows of ATP synthase on top of it. They also perform subtomograph averaging to resolve the domain organization that is responsible for the formation of the dimer, and then how the ATP synthase were able to form the 2 arrays, and how the formation of the 2 arrays actually generated a very unique helical and tubular structure of the membrane cristae. So, from this type of study, many of them during those period of time, first of all, they showed our effort to apply crowd-ET in cell biology to understand protein complexes and organelle organizations in close to native state. At the same time, it actually demonstrated the limitation because we were unable to study the real in situ biology, again, because of limitation sample thickness. So next one, it comes the FIP. So it's about 10 years ago, and then FIP application becomes feasible in sample prep. So basically, that application completely opened up the inner world of the cell, eukaryotic cells, for crowd-ET study. So I'm not going to go into technical details because you will have talks on this topic. I'm only going to show one example. This is the nuclear peripheral region by crowd-ET of cell lamella generated by FIP milling. This is the pillar cell. So basically, you will be able to resolve the membrane envelope nuclear complex and also all the ribosomes in translation very close to the nuclear peripheral regions. So those structures used to be very deeply hidden in the cell plasma that are not accessible by tomography until application of FIP milling. Right? And also, I also want to discuss a little bit of subtomograph averaging because, as I mentioned, crowd-ET is a very powerful tool to understand the polymorphic structure, but very often they are recurring elements in the tomograms. For example, ribosomes in a tomogram of a cell or the capsid in the tomogram of the virions. So for those recurring elements, we can actually pick them out, align them, and reconstruct them to get a subtomogram average so that we can get higher resolution, better understanding of those recurrent elements. So, but if you look at the resolution currently achieved by subtomograph averaging and compare it to single particle analysis, you can see there's a dramatic difference. So for subtomograph averaging nowadays, for the maps deposited in EMDB, basically they're still in the nanometer range from about 10 astrons all the way to 40 to 50 astrons. But for single particle analysis, look at the distribution. For well-behaving samples, it almost become a routine for you to get like a 3 to 5 astron resolution, right? And then, but I want to get your attention to one of the really well-behaved samples for apple ferret. We know this is the model sample we use for resolution assessment in EM, crowd-EM. So by subtomograph averaging, you can actually get to about 1.6 astron resolution. And then very comparable, in a comparable range to the best resolution you can get by single particle analysis. I think for this comparison, my conclusion is the subtomograph averaging data processing is well established. Currently, the limitation is really either the sample itself or the sample preparation. Right? So for subtomograph averaging, I also want to emphasize the pioneer work from Julian Muhammad's group. So about a couple years ago, they started to apply crowd-ET to the entire mycoplasma cells, and they were able to extract subtomograms of ribosome and to perform subtomograph averaging. In a recent study, they were able to use antibiotics to treat the cell, and then do subtomograph averaging of ribosome from the treated cells, and then able to resolve the resolution from subtomograph averaging up to 2.9 astron resolution. This is a very nice resolution for you to be able to resolve all the side chains and also the antibiotics to understand how the antibiotics inhibit function of ribosome at different levels in real in situ situation conditions. Okay. So I think, the introduction to tomography is not complete without a small discussion of AlphaFold or structural prediction. So what is the impact of structural prediction in application of tomography? So I'm going to use just one example that many of you, especially local people, should be really familiar with. This is the human nuclear complex structure resolved by integrative structural biology. So we all know, the nuclear complex is a very challenging sample. First of all, it's really large. It's probably the largest structure in a cell. And then also it has 1,000 subunits. Present them as multiple copies of a set of nucleoporin proteins, complex structure. And also it's also dynamic, very dynamic. So it basically goes through the dilation and constriction motion all the time. So they were able to resolve the structure first by tomography, taking tomogram, resolve the structure of the dilated and the constricted states, low resolution. And then by applying structural prediction, they were able to get structural models of the neuroporin structures or subcomplexes, and then validated by benchmarking against available X-ray and crowd-EM structures. And then the applied integrated modeling basically fitted those structures into the envelope generated by crowd-ET. So basically, I want to end the introduction to tomography using this sample to show all the recent development in structural prediction really can accelerate application of crowd-ET to the next level and compensate for the low resolution status of the technology. So basically the first part of the introduction, really, I just want to deliver the message. So when you're doing your thesis, when you're doing your project, when do you want to use CrowdET? If you want to look at the unique feature of your sample, your protein complex, or you want to understand the dynamics and organization of your favorite sample when they're still inside the cell. And then also subtomogram averaging really provided an alternative for single particle analysis in certain situations. Okay, any questions so far for the first part? Okay, so for the next 10 minutes, I want to discuss my research, use it as one example to a real life example to show you the workflow of CrowdET application in cell biology. So in my lab, one of the projects, we're interested to understand drug molecular mechanism underlying antifungal drug action. So because fungal infections, they're very costly and they pose health risk, especially to immunocompromised individuals. So, current FDA-approved drugs target unique feature in fungal cell. Target number one is called ergastrol. It's basically the equivalent of cholesterol in a human in the plasma membrane. And then we have proline and azoles against the lipid composition. We also have another class called echinocandins. It targets the glucan composition in the unique cell wall. Glucan is synthesized by glucan synthase. And glucan synthase has two subunits. The larger one is a membrane-embedded unit and also has a regulatory subunit. So echinocandin drugs target the larger membrane-embedded subunit. And then the first 3D structure, high-resolution structure, was resolved by CrowdEM single particle analysis in 2023, still pretty recently. So this structure is about 3.4-ish atom resolution. So it has a lot of nice features, enough detail to show you the active site and also the channel formed by the transmembrane domains so that glucan can be extruded from the cytosolic side to the extracellular side. It also resolved several very unique folds in this protein complex. So why we still want to do tomography to understand the protein complex? First of all, all those protein complexes, they're purified by detergent. And then, the same protein complex from the same species purified by different detergent, they actually show slight conformational changes at the active site. And also, those complexes purified by different detergent, they also showed different susceptibility to echinocandin drug. CSF is called caspofungin, is one member of the drug. And then some of them, by some detergent, actually showed increased activity instead of decreased activity. That tells maybe the native lipid environment actually is involved in the drug action. If you go back to the structure again, the high resolution structure, we know in clinical settings, actually the resistance to the drug involved the mutations in 3 hotspot regions of the protein complex. And then in the high resolution structure, the hotspot regions happen to be where you can find all the integral lipid molecules that further support the idea that resolving the structure in the native state will help us to understand the molecular mechanism of drug action. So we did tomography of isolated membrane. So instead of isolating or purify the protein complex, tomography study allow you to just isolate the membrane and then also going through minimal processing so that the complex is still in its native membrane embedded state. So tomogram, you see lots of black dots. Many of the black dots are membrane protein complexes. You also have some of the cytosolic composition left in the sample prep. Okay, I'm just gonna. And then because compared to single particle analysis, crowd ET projects feel purely, we call it information rich. In another word, called very noisy. So you have to figure out what's in your sample, right? So very often we perform, proteomic analysis to figure out the potential candidates in your tomogram. And then also you have to go through some cleaning up process based on the cellular location, sizes, and the abundance to give you a reasonable list of potential players in your tomogram. In my case, glucan synthase, FKS1, turned out to be one of the top runners in the sample. And also the second one, more abundant one, is PMA1, it's a proton pump. From there, we're gonna do annotation. You're also gonna have a tutorial, a hands-on session on annotation. This one I did it in collaboration with Moyan, so we use the Eman2 as a platform that you can annotate major structures in the tomogram, including some of the cytosolic components. And then the donuts are the proton pump, and then also the little blue dots are glucan synthase, our targets. So from there, after annotation, you will be able to have all the coordinates of your interested target. From there, you can do many things. For example, you can extract your target for subtomogram averaging. And then also based on those coordinates, you can do distribution analysis and the clustering analysis to understand their functional distribution of states in cellular settings. And of course, you can do tomography of drug-treated membrane, and then going through the same process, proteomic analysis, and also annotation for you to better understand the structure and distribution dynamics of your protein complex after treatment. Right. And then the idea would be you want to compare after treatment versus before treatment. And then really the logic thing to do next will be subtomogram averaging of glucan synthase from the treated membrane versus the untreated membrane. But unfortunately, the resolution we can achieve at this point is lower than we needed to resolve the drug density or changes to the integral membrane network. Nevertheless, from the two pictures, you can clearly see there are some changes in the organization of microdomain reflected by the loss of clustering from the donuts. From those, you can kind of propose new ideas. For example, we think in this particular case, those integral membrane, integral lipid molecules probably involved in the drug complex interaction. And then when the drug inserted into the membrane, very likely disrupted the local lipid network reflected as changes of the membrane microdomain. Once you have a model from what you observed from the tomography study, you need to test it and validate it. And the one tool we often use is nucleogenesis. In our case, we generate deletion to one of the genes involved in sphingolipid biogenesis. So by doing that, the mutant will have defect in sphingolipid biosynthesis and changed lipid composition. And then first of all, you can also apply, for example, biophysical tools. This is called micropipette aspiration. So we basically use a micropipette to suck the spheroplast from the mutant. And then we quantify the membrane deformation in response to the aspiration pressure, and then compare it to the wild type. It shows that the mutant in the displayed that demonstrated some change membrane property consistent with this change the lipid composition. And then, of course, you can do the drag test of the mutant, and apparently it's more resistant compared to the wild type, supporting our models. Okay. So from this, the second part of my talk, by showing one project, the workflow, the message I wanted to deliver is probably electron tomography is actually a really powerful tool for you to visualize that biological process, what is happening in the cell, to resolve the structure of your favorite protein complex in its native state. And then when it's combined with complementary approaches, such as mutagenesis, protein chemistry, mass spec, and biophysical analysis, it becomes even more powerful to build your model and biology story. That's all for today, and I have to acknowledge key players contributed to the little story I shared at the second part. And you can start. Any questions? Okay.
I'll start off. So you were talking about saying when to use electron tomography. Are there particular targets or compartments within the cell that you find that tomography works better versus other biophysical techniques, for example, single particle?
That's very hard to say without basically people being focusing on, for example, plasma membrane protein, class membrane regions, or self-referential regions. And then now with FLIP, we can basically go deeper into the cell plasm, even go into the nucleus. But still, you have to consider throughput. FLIP milling still a very low throughput. It's the bottleneck of the whole workflow at this point. So, but again, with split milling, eventually you will have access to every single corner of the cell. But you also have to consider how many data you need to build a story. Either you basically pushing for throughput of the workflow, or you have to rely on supplementary or complementary approaches to build a story. So depending on what you want to work. And abundance is really another factor you have to consider. You don't want to spend your days just looking for one case of your complex.
That means if there is low abundance complex, then do the single part.
That's okay. Then you basically want to scale up your culture and then try really hard to purify it to get to go through single particle. There are other ways. You can also overexpress it.
Yeah.
And then you can also label it so that you have an easier job finding it. The other fits and potentially use.
So you were saying that tomography data is very information rich, it's noisy. How do you like to validate if it is very noisy that you are actually looking at the right targets for just validation in general? What are your tips?
So there are different ways, or there are easier ways, there are harder ways, right? Some of the target, they have very strong structure signature. For example, mitochondria, ATP synthase, microtubule, you basically see it and you know it. And then there are other features that you don't have a clear picture of what they look. And then the good news is now you have AlphaFlow. You kind of know what kind of shape you're looking for. And also they're correlated by the electron microscopy. You basically can add a tag to it. You can overexpress it. You can also do deletion, basically mutagenesis to play with it. If you delete it, that part of the density is gone. Very likely that's your target. Or you add a tag to it, fluorescence, you'll find a cluster of the fluorescence signal. Very likely that's where the target is. And also eventually, when you think you eventually find it, you still need to validate it. For example, one way I can think of is you do subtotal averaging. You can eventually get a structure just like the one by single particle analysis deposit in the map, and that's a great validation. Right? Yes.
The resolution limits. You have an example of the ribosome 0.9. how good can we possibly get, given that you have the amount of particles and the sample thickness, how good can we expect a good resolution to be under ideal conditions?
So are you talking, so what determines resolution by subtomogram averaging, right? Hartman number is definitely one, and also sizes, and also symmetry, right? So that one I show is 2.9, but remember, that's actually an intracellular structure. The ribosome is not purified. They are extracted from tomograms of intact cells, which means the neighborhood are very crowded, very noisy. So considering that condition, I would say 2.9 is very impressive already. But let's say you can purify ribosome and then just smear them on the EM grid. Very likely you can get better than that, simply because the contrast, ice thickness will be much lower. And also, as I showed the apophyllite example, the resolution is 1.6. The highest that you can get from by single particle analysis of the same sample is 1.2. So I would say they are very comparable at this point. That's why I said from that example, I think currently the limiting factor is not data processing. It's more likely to be your sample and sample package.